Good afternoon and welcome to our front row lecture series. I'm Donna Blackman. I'm chair of the chemistry department at the California campus of Scripps Research. As you may know, we have two campuses, this campus here in La Jolla and our sister campus in Jupiter, Florida. And our speaker today is a member of the chemistry department at our Florida campus. And we're thrilled to have him here because he is an exceptional chemist and a translational scientist. So let me tell you a little bit about Matt's background. He earned his a BS in chemistry from the University of Maryland and went on to do a PhD at the University of Rochester. After postdoctoral studies at the Swiss Federal Institute of Technology in Zurich, he came back and started his independent career at the State University of New York at Buffalo, which is also known as sunny Buffalo. But we are delighted that we were able to lure him away to sunnier climes at Scripps Research. So Matt Disney doesn't just invent new medicines, he invents entirely new ways of treating disease. In fact, he's creating drugs for diseases that have been traditionally thought to be completely undruggable. These include Huntington's disease, myotonic dystrophy, and spinal muscular atrophy, among many others. These are diseases that affect millions of people around the world, and there are no satisfactory treatments yet. But Matt has pioneered a new method for targeting these diseases. And he'll explain this to you, but most drugs on the market today focus on proteins. But he's developed a method that focuses on RNA. And RNA is the cellular machinery that helps to build the proteins. And this is an exciting new technology that has a great potential, which is still not met and fairly untapped. So Matt's accomplishments in this work have not gone unnoticed. Earlier this year, he was awarded the Sackler Prize, which is a very prestigious international award granted to scientists under the age of 45 who have made outstanding contributions to their field. He's received numerous other awards, including the NIH Director's Pioneer Award, which supports scientists of exceptional creativity. I know you're going to enjoy his talk. He's one of our most enthusiastic and passionate scientists. But before he begins, I just want to take a minute to thank you, because many of you in the audience, I know, support our research. Scripps Research is a very special place. We are ranked the #1 nonprofit research institution in the country, both for the quality of our research and for the impact of our research. And something else that really sets Scripps apart from other institutes is that we have one of the top graduate education programs in the world. We produce scientists who go on to tackle problems of human health and find cures for diseases, working at top universities, research institutes, and companies all across the US and around the world. So this is highlighted in our mission, which is to advance scientific understanding, educate the scientists of tomorrow, and impact human health across the globe. So we accomplish that every day because of your generosity, so thank you. So following Matt's presentation, there'll be a reception in the courtyard. We'd like you to join us for that. But for now, let's please welcome Matt Disney.
Thanks, Donna, for that great introduction. Donna admitted that she couldn't find dirt on me, and she looked. My retort was she didn't try very hard. As Donna mentioned, I started my academic career at a large state institution that gave me a chance to do the research that we were very passionate about. And about 4 years in to trying to answer fundamental questions about molecular recognition, which is basically which molecules like to stick to each other. So after trying to figure out if RNA would stick to small molecules or the medicines that we like to take by tablets, I got a call by Jerry Joyce in my office one time. And Jerry was the dean of faculty at Scripps at the time. Now he runs GNF, and he's a professor at Salk Institute. And so Jerry offered me a chance to move to Scripps, Florida. So I jumped at that chance, despite the fact that after my first visit, I came back and we found out that my wife was 5 months pregnant, which was very interesting. I wondered why my wife was figuring this out at 5 months. Full disclosure, she is a scientist, so it made it all the more unusual. Though admittedly, I could have figured this out myself. And so, I went from an institution that paid all of my salary to go to Scripps. That's one that we call on soft money. And so what Scripps does is it gives you an opportunity to raise your salary by getting grants and doing science. And so, Jerry, when he called me to walk through the offer, I didn't really listen to him. And luckily, Jerry can be a bit verbose at times and talk. So I was able to take the phone away from my face and catch my breath a little bit. And of course, we were going to move to Scripps. I'm one of 7 kids, and my dad and mom invested in all of us. And I thought, when I talked to my dad about this, he said, looked up Scripps, and he goes, "Hey, Matt, wow, this is incredible. I could probably get a major return on all the investment that I've made in you and the other kids just by you." So it was pretty much a no-brainer. All right, so I'm a representative of John and Hope Disney, but also the Disney Group at Scripps, Florida. And so I think we're really, Donna touched on this, we're really blessed to work in an environment. Not only do we have top-notch scientists that are professors that you know about now, we also work amongst scientists that you're going to hear about in the not-so-too-distant future. So if we go to a pharmaceutical company, the beautiful thing about Scripps is they tend to talk about Scripps faculty members in their first name. So Donna is Donna Blackman, Phil's Phil Barron, Dale's Dale Boger, Pete is Pete Schultz, Ben is Ben Cravat, Jeff is Jeff Kelly, Hugh is Hugh Rosen, Disney is Walt Disney. So people that I work with in the lab are, so my lab's about 20 people. That is, we have undergraduates in the lab, we have 2 high school students. People that I'm going to talk about today include Ali Angibello, who did work on targeting the RNA associated with muscular dystrophy, who worked with Suzanne Zuchek. I'm also going to, who's right here is Ali. Suzanne is over here. I'm going to talk about work that Matt Costellas and Tanya Khan did on trying to develop molecules that would target RNAs associated with cancer. And I'm going to talk about work that me and Sai Velagapudi did to try to develop a platform that could drug RNA broadly with small molecules. And we really want to be grateful to all of you because it's pretty amazing to live in a country where you can have ideas at the middle of the night, and you can come into the lab and someone will give you funds to be able to test those ideas out, right? You can get grants. We all bemoan as scientists about having to write grants. But how amazing is it that your ideas, you don't get a contract, you get a grant. So if you have a new and better idea than what you got funded for, you can do that. That is pretty amazing. And that's because people like you pay your taxes, hopefully. And it's also because people like you give to places like Scripps Research. So I'm very honored to talk to you today. Okay, so I'm trying to boil down into one sentence what we've been doing. And we've been working in the area of RNA molecular recognition for 15 years. It's become really hot in the past several years. There's been probably 10 companies that have spurned out to work in this area. That's probably on average got about $500 billion or $500 million of funding. And so what we sought out to do was study the basic science of how RNA folds and bind small molecules to develop new approaches to potentially treat diseases. And I am trained as an RNA biophysical chemist. That is, I'm a person that looks at RNA structure and tries to figure out how it folds. And so, a lot of science is about history and about how people viewed things at one point in time, and then we need to be recognizing how they view it today. And so I went back to the 70s, and so there's something called the central dogma that people think about in biology, which is DNA goes to RNA, goes to a protein. And so this is the view of how biology worked from a biologist or a chemical biologist in the 1970s, is that information transfer would go from DNA, that we all are very sensitive and know what it is, it's our genetic material, that would be transcribed into RNA, namely messenger RNAs, and those messenger RNAs would be decoded by a ribosomal protein complex called the ribosome to make our proteins. And I can tell you that now we don't think about biology in this way, is that in the middle of this slide is RNA, but one thing I'm going to tell you today is that really the central biomolecule in all of this biology is RNA. And so if we think about scientists today, even today, that's thinking about how to discover small molecule medicines or things that you swallow as a pill, like aspirin or ibuprofen, how they think about things is they think about DNA and they go, okay, DNA, chemotherapeutics target this. So many chemotherapeutics that are used to treat cancer in a not so non-specific way actually react with DNA, and they're built from chemical weapons that were used to kill soldiers in World War I. On the far right of the screen is people, how they think about proteins, is they go, I kind of know how to target this. So I'm really comfortable in, if I have a protein that's modulating a pathway, doing high throughput screening or various other approaches that we have to figure out small molecules that can bind to that protein and affect its biological function. However, for RNA in the middle, they really look at it and they go, I have no idea what to do with this. I don't, but I know it's really important. And so that's where the void that we like to work in. So I, by my nature of being one of 7 kids, am a very competitive person. However, what I wanted to do when I started a lab is I am so competitive, I do not want to do what other people are doing. And I think if there's 10 people working in a field, part of the beauty of coming to Scripps is they almost expect you to work where there's dark spaces in science that you think is shining light on it can have big impacts. And so that's what we tried to do. So why do people think about protein-targeted small molecules so centrally in the drug discovery work that's done today? Well, the reason that they do that is that proteins, as many of you know, are built of amino acid building blocks that have very diverse folds in them. So amino acids can be charged. They can have positive and negative charges. They can be grease balls. They can be soluble in water. They can be, generally, their side chains can be insoluble in water. And when you build a biomolecule based on diverse starting materials, well then the molecules that emerge from that can have very significant diversity based on the building blocks that they're composed of. And so that diversity can cause proteins to have very complicated structures. And some of the complicated structures are shown here on this slide. And what I've done is I've carved off the front section of a protein, and you can see in the center of some of these things, my laser pointer, which is really not working, is making this harder on me. But you can see here's a small molecule that's stuck on a protein in the center for a compound that's used to treat heart disease, and here's a small molecule that's stuck on another protein that's associated with cancer. This molecule on the far left is a compound called Gleevec, and it's a very interesting molecule in the history of medicine. What was found in blood cancers is that patients can have a chromosomal rearrangement. That is, they can have something called a Philadelphia chromosome, where part of their DNA breaks off, 2 parts of your DNA get broken off, and they reattach each other in the completely wrong way. The analogy for that is, coming from a large family, my mom would have a few Hummels around the house, and we decide to throw baseballs in the house, we'd break one. Well, mom could figure out which one was broken by nature of us not gluing it properly back together. And so that's how the Philadelphia chromosome is made, and it basically puts a cancer-causing protein in a position on a chromosome where it's constantly being made. And so that was discovered in 1960. It only took 41 years for the compound that targets that protein called Gleevec to be FDA approved. And what that did is it took patients that had a blood cancer that were going to die within 18 months and gave them life. So they would be able to survive much longer than 18 months, in some cases decades with that disease, until resistance inevitably came up. And this Gleevec is part of the revolution in precision medicine that we're living in today. It spurned it on. It's what has initiated what's called targeted therapeutics. And so as you can imagine, the way that proteins bind to small molecule medicines like Gleevec, if it's much like how a key reads out a lock, that is, a small molecule bind to a very specific protein by reading out all the ridges and edges in that protein and bind to it. Much like a key will unlock the very specific lock that it's meant to bind, a drug will read out the very specific pockets in a protein that it is meant to be a medicine against. One thing that has revolutionized discovery, and I don't have to convince you of this in principle, has been genome sequencing. And what's shown here is a plot of the cost per genome that we've had throughout the past 2 decades in bioscience research. And so what you can see is now we're at a stage, and this data is from the NIH in 2016, where we can sequence a whole genome for about $1,000. That's pretty amazing. What makes it all the more amazing is what I've also put on this plot is Moore's Law. So Moore's Law is computational power of computer chips will double every few years. And you can see that's a pretty consistent rate. The efficiency by which we've been able to sequence genomes is far outpacing that. So what are some consequences? The computational power on your iPhone in the calculation app, the calculator app, is about 10 times more than the computational power that got a man on the moon. The computational power that we have on our wrist, if we have a Fitbit or an iWatch, and we have in our pocket, if we have a Samsung device or Apple phone, is about 100,000 times the computational power that got a person on the moon. And DNA sequencing efficiency is far beating that. That is amazing to me. Science is amazing. One of the main things that came out of the first draft of the human genome sequence that happened in 2001, many of you will remember there was an announcement. President Clinton had the NIH consortium and the private and public sector had an announcement that the draft genome sequence was coming out in 2001. There are many surprises in that genome sequence. One was that the number of genes that humans have are much less, about 10 times less than what wheat has, right? We can all agree that humans are much more complex than wheat, I hope. And so the consequence of that is if you look deeper into that information, it turned out that humans actually only 1% of our genome actually encoded for protein, but about 80% of our genome made RNA. And so those RNAs didn't necessarily have to encode for protein. They could have what's called long non-coding RNAs or be non-coding RNAs that would have very specific functions. Non-coding RNAs, for example, can cause developmental timing. So as you have embryos, how they switch from different types of embryos to develop into an animal can be triggered by your RNA. Your RNA can switch states in how we would, as you're a child inside your mother, how do you breathe? Well, what you do is you form a version of hemoglobin that binds oxygen with a higher affinity than the oxygen that the hemoglobin that your mother carries, and that's how you transfer oxygen through the umbilical cord. And so RNA and non-coding RNAs played a really essential role. And if we look through organisms, actually organismal complexity scales with long non-coding RNAs. What does that mean? If I want to look at the difference between me and a mouse or a cat or a dog, I don't want to look at the protein coding genes. They're going to all be very similar. What I want to look at is the non-coding RNAs, is that the differences between species are going to be most driven by differences in non-coding RNAs. So what does this have to do with druggability, which is what I've been working on? Well, I'm going to go back to what I said earlier. It turns out that the amount of protein made from a human genome, about 15% of that is considered to be drugged. And so what that means is people use terms like druggable and undruggable. Kim Janda hates this if Kim is in the audience. So I refer to not say, hi Kim, I refer to say not yet drugged, right? Is that a better term? No, okay. And so what we do is there's about 15% of our proteins have been drugged, and people are working tirelessly at Scripps, like Ben Cravat and other people, to try to figure out which of the rest of the human proteome can be drugged with a small molecule. In contrast, if you look at human RNAs, 0 of them have been drugged with a small molecule, none of them. But I just told you that human RNAs are very important molecules in terms of understanding organism differences. And the other advantage of RNA is that it can be amplified and very rapidly sequenced by using similar technologies that were used to sequence the human genome. And also, RNA plays key roles in essentially all diseases. So every disease that ranges from an influenza viral infection to if you have multiple sclerosis, to if you have ALS, to muscular dystrophy, to Huntington's disease, to cancer, to fibrosis, dementia, can all be caused by an RNA. There's an RNA target in each one of those diseases. And what you'll see on the right of this plot is that the number of publications in RNA and disease is just exponentially growing as a function of time. And what's shown in that word plot are some of the major diseases that are associated with RNA. Okay, so if I were to go to, probably 10 years ago, and I have a slide. It actually looks pretty bad where I give the reviews of my NIH grants. They just basically said we couldn't do anything that we were proposing to do, which is, that's how it always goes, right? But I think scientists are somebody asked me, oh, how do scientists, 90% of what you do is always failure. How do you keep going? Well, so I think scientists are some of the probably the most optimistic people that you ever meet because they actually love failure. It's not really a failure, it's science, right? If you keep failing and something doesn't work, well, that's gonna lead you to understand the system and figure out something that will work. So skeptics in some of those initial grants would say that RNA is not druggable because it doesn't fold. It doesn't fold in a complicated way like the proteins do that are made of 20 diverse amino acids. RNA, much like DNA, is made of 4 bases. And those bases, which are shown to the left here, are boring. They all look the same. They don't have a positive charge. They don't have a negative charge. They're not, a few of them aren't grease balls, and a few don't dissolve in water. And furthermore, they don't form these elaborate structures. They just form the base pairs that are on the right. And so that's the equivalent of looking at and trying to figure out a way to climb El Capitan. So if you look at that, and RNA has these very non-diverse structures, well, it's basically trying to drug RNA is like trying to get something that sticks right in the middle of this solid stone that's just emerging from the earth. There's not these ridges and crevices, or the thought is within RNA, like there are proteins. And so there's not going to be places where you can hold onto it and grab onto it to drug it and target it with a small molecule that's very specific. And so just like people have climbed this mountain without ropes. This is crazy, by the way. I'm getting sick, like motion sick, looking at this picture. If you use your brain, right? If you think really hard and you train really hard, you can drug RNA and you can climb El Capitan. Some of the crevices aren't so big, but if you figure out ways to stick your hand in and pull yourself up, you may be able to pull yourself all the way up to the top. So here's how RNA really folds. So RNA is a single-stranded biopolymer. It's not like DNA. So the image that I showed you where every A pairs with a U and every G pairs with a C is wrong. So RNA is a single strand, and it folds onto itself to form canonical base pairs, but also these looped regions. And then RNA will also fold to the far right in what's called a three-dimensional structure. But really, that secondary structure is a way that we draw and most easily represent some of that 3D structure in 2D space on a sheet of a page. And what we also know is that RNAs not only contribute to every disease, but they have very robust and different foldings that they can have within each one of these RNA targets. So here's just 5 random RNAs that I pulled off the page. So to the far left is an RNA that's associated with dementia. To the far, let me see. Oh, maybe it works. Oh, is this going back? Oh, yeah, almost. Here we go. Oh, it works now. What did I do in the beginning? All right, so this is an RNA that's associated with dementia. This is an RNA that's associated with Parkinsonism. This is an RNA that's associated with cancer. This is an RNA with heart disease, and this is an RNA with ALS. You can see they have holes and crevices in them, and those holes and crevices can be filled with small molecule drugs. Okay, so what did we do to start on this journey? So I got my PhD from the best scientist I know is a guy named Doug Turner. And he was a guy at the University of Rochester, and our labs are in the basement, so he liked to be by himself. He's the nicest guy I ever met, frankly. Dale's close. All right, Dale's nicer. Only because Dale will watch this on YouTube, and I'm not sure Doug will. And so Doug did things like measure the energies of a hydrogen bond, and he basically developed a computer algorithm where you could go and look at RNA sequence and determine which of the RNAs would fold like structures like I'm showing here, and do it from using entire genome sequence. And so I thought it would be pretty cool if we could use this software to identify if there were sites in an RNA that were folded. And then if we could figure out those little crevices in the RNA, if we could figure out which of those bound to a small molecule drug. And if we could do that, in some sense, we'd be able to decode the entire set of RNAs that are made by a human genome with small molecules that could bind to these RNAs and affect their functions. And so the basic scientific question that we set out to ask and answer was, are there RNA folds that bind small molecules, and what are they? Are there small molecules that bind to RNA folds, and what are they? And the way that we went about doing this is we took a drug, which I'm schematizing here in this yellow dot, and we tested it we developed a method where we could test this dot for binding to hundreds of thousands of RNA folds simultaneously to find the one that it perfectly fit with. And we could do this in a massively parallel way. And so what that means is I could screen libraries or thousands of drugs for binding to thousands of RNA folds simultaneously. And when we do this, the drug would pick out a precise three-dimensional fold within that RNA structure. And we got so good at this, we could make tables and tables and tables of this information. And what it allowed us to do is we could define things like there are drugs that bind RNA. We know that actually known drugs that you take every day bind RNA, but people never thought it was druggable, and therefore they never thought that it could affect the function of these RNAs. And so we're doing things that's called repurposing of drugs. So we work with Pete Schultz and Caliber and Arnab Chatterjee over there, who have taken basically every drug that's ever been put into man and shown to be non-toxic. And so we're testing them to see which of these drugs bind RNA. And I can tell you, many drugs that are not only FDA approved, but failed through the approval process actually bind very specific RNAs very potently. And we can use this to not only identify new targets for these small molecules that are not protein-based but are RNA-based, and also define why they failed in a clinical trial, because some of these molecules can be toxic. And we think that some of the toxicities within these compounds can be due to binding to RNA, but yet people never thought that RNA could bind these drugs and didn't look at them. And so we have these tables of information. What we're able to do is, and I'm going to show you this in the next few slides, is basically make a platform that allows us to decode the human genome or the human transcriptome for small molecule binders. This is an Enigma machine. And so an Enigma machine was used to decode messages that the Germans sent in World War II by a guy named Alan Turing, who was a British scientist who developed a lot of the early algorithms to use computers. And so basically what we did is we used our information to basically identify and develop an Enigma machine where we want to basically ambitiously break the entire code, and we call this approach Inforna. So that's what we're doing. Here's how Inforna works. So scientists always stand on the shoulders of other scientists, and so I'm standing on the shoulders of work by people like my graduate advisor, Doug Turner, people like Michael Zucker and Nacho Tinoco, even guys like Paul Dotty. And so what you can do is you can take RNA sequence, which is plentiful, and you can take it and identify which regions in a specific RNA are folded. So here's what an RNA looks like in a schematized way. So these are regions of the RNA structure that have a high probability of folding, and these are regions that are unfolded. Now, I do realize I'm in San Diego, and Ionis is right up the street in Carlsbad. Ionis is a company that uses pieces of DNA to bind to RNA and to degrade them, and they've had many successes in making therapeutics. What they do is they focus on regions of RNA that are unstructured. What I am focusing on regions of RNA that are highly structured to design small molecules that bind these RNAs by using sequence. And so once we identify there's a highly probable fold within an RNA, we can go, oh, is this a druggable pocket? And how we define a druggable pocket is we look at our Enigma machine. Again, I told you that we were selecting drugs that bind to RNA and what the folds are. And so we can query a specific fold that has a three-dimensional shape and go, here's a small molecule that'll bind to one or two sites in that RNA, and we can very rapidly and rationally identify that by using the RNA sequence. If that binding compound itself can affect the biology of an RNA, but another way that we can affect RNA's biology is by making these molecules into cleaving compounds that bind to and destroy transcripts or RNAs in cells and animals. Now, this Inforna has some subtleties associated with it that I want to take a little bit of time to describe to you before I talk about 3 applications of this approach. So one thing it allows us to do, and all of us take medicine, we don't want a medicine that binds to many, many targets, because if it binds to many, many targets, it's not going to be very selective. And so what Inforna will tell us is a specific molecule. Does it bind to many targets? Does it bind to every target? Does it bind to a target that will kill every cell, which you don't want, right? If it does that, you throw it out, okay? What it also tells us is if a molecule binds to a few targets, what are the on targets and what are the off targets? And so it'll say, oh, this molecule binds to this off target and this on target, and we can rapidly define ways in which we can accentuate the molecule for binding to the on target and eliminate it binding to the off target to make molecules very selective in a very rational and predictable way. The other thing that it can allow us to define are molecules that bind to RNAs where it's very selective and the site that the compounds are binding to the RNA are unique. And this can get very important because it's like hitting, it's like getting a bullseye on playing darts. I don't play darts, but I guess people hit bullseyes. And so this will tell us that this is a unique and a functional target that'll bind to an RNA. Okay, so we took this machine and we broadly displayed it across the human transcriptome, and we identified a wide variety of RNAs that were causing disease that we could drug with small molecules in this very rational way. So one case, and I'm going to talk about two cases here. So one case was cancer. And so all of us, I think, are affected with cancer, but specifically what we found is small molecules that would target RNAs that are associated with breast cancer in two ways. Breast cancer it can be separated into two broad types of breast cancer. One is called triple-negative breast cancer, and one is non-triple-negative breast cancer. So if you have triple-negative breast cancer, that molecule has no targeted therapeutic for it. That is, there's no approved medicine that'll target the genetic signature in that cancer that'll kill it specifically. And so patients that unfortunately have triple-negative breast cancer, their treatment options are radiation, surgery, or basically medicines that are chemotherapeutics that are repurposed from World War II medicine. So these are not very good, and they're not certainly targeted therapeutics. And so what we did is we used Inforna to identify very upregulated and associated gene products in triple-negative breast cancer that what they do is they suppress apoptosis. So when you have cancer, you get basically cells that overgrow many, many, many times. And the way that they overgrow or continuing to grow is they suppress what's something called programmed cell death or apoptosis. And so those RNAs that continue to allow cancer to suppress apoptosis would be good targets, because if you could silence them, you can trigger these cancer cells into killing themselves. And so what Inforna did is it identified this small molecule in blue and this other small molecule in orange for binding to an oncogenic or a cancer-causing non-coding RNA. We then could link these two compounds together that would target this non-coding RNA very specifically and with very high affinity and would be very potent. When we delivered the two halves of these molecules into these triple-negative breast cancer cells, what's shown here is a plot of percentage of cells that are undergoing death, or this apoptosis when they're given our drug. The two halves of the molecules have no effect on these cells. Interestingly, if we take healthy cells do not express this target. The molecule also has no effect if we take healthy breast cells or any number of healthy cells. That molecule, however, at a very low dose is able to cause 90% of these triple-negative breast cancer cells to undergo apoptosis or to kill themselves. We could then take these molecules without any lead optimization. And so Inforna, at least at this level, this is called a chemical probe, I'm going to talk about that at the end, is so good that we could take what's hot off the press and throw it in an animal. We don't have to do typically molecule, we get a lead and you do a lot of medicinal chemistry to see if it works in an animal because you're worried that the animal will degrade the compound or it won't get to the tissues. Inforna is so good, it can tell us which compounds to make. It can tell us one compound. So we made one compound. And then we can deliver it in a mouse, and this is basically a tumor that has imaging on it, that you can image a tumor, and you can see when we deliver this molecule in a mouse, it'll slow the tumor growth. Another issue that patients can have when they have breast cancer is something called metastasis, and that's actually a cause of death in cancer patients, many cancer patients. And so what happens is cancer, when it grows, it's hungry, so it tends to have a lot of blood going through it so it can feed the tumor. And what happens is over time is because it has this vasculature, this metastatic cancer will start to slough off cancer cells in the blood. And the consequence of that, if you have breast cancer that's untreated, is you'll get breast cancer initially, but then it can go to your lung or it can go to your brain because those tumors are becoming metastatic and they're basically going to these other organs and sitting there and growing. And it turns out that it's known that many RNA genes are upregulated to cause cancer to not only be cancer, but to also become metastatic. And some of these metastatic cancers you can detect from a patient's blood. And so what we did is we designed a molecule that can target these RNAs that are upregulated and causative of metastatic cancer by using Inforna. So basically we identified a compound in yellow that would bind to two sites. This is a non-coding RNA that causes metastasis of breast cancer to the lung. And here's a lung of a mouse. And you can see what's on the left is each one of these spots are metastatic tumors in a lung from a mouse that's delivered this metastatic breast cancer. When we give it this compound, you don't see any of these nodules in the lung. And we're making like 1 or 2 compounds here. We're not doing high throughput screens of hundreds of thousands of molecules. So that's cancer. I also want to talk about rare diseases. My niece has Prader-Willi syndrome. And so I have it's a rare disease where you're missing snow RNA. So it's the syndrome where it's my first niece or nephew, very special young lady. And so I have a very high affiliation and gravitational pull. This is all gravitational pull. The molecules are telling us what they want to target. We're not imposing a target on them. We're just riding energy waves. The molecules are going, oh, we want to stick to this. And we're going, oh, you stick to that, that's in cancer. So that's the way the calculation is set up. These are some very important diseases that we have insights into drugging by using Inforna. One is called Fragile X syndrome. That's the only single gene cause of autism. Okay, it affects 1 in 4,000 patients. One is called Fragile X-associated tremor ataxia, which is the most common adult onset form of tremor. And so what happens is the grandfather and the grandson actually have the same RNA repeat. These are RNA repeat diseases. It's just that the grandfather has a shorter repeat and the grandson has a longer repeat. So that tremor goes to autism. The other thing is you can have what's called myotonic dystrophy. That's the most common adult onset form of muscular dystrophy that's caused by an RNA repeat. Also ALS, the most common form of ALS. And ALS is linked to frontotemporal dementia is due to a humongous repeat of G4C2 that's in a non-coding RNA. And that was discovered in 2011. I also want to spend some time to talk about Huntington's disease. So I'm not going to talk about Huntington's disease, but I want to talk about this person in this picture. And this is some of the things that happen in the rare disease space. This is Nancy Wexler. Nancy Wexler's dad was a guy named Milton Wexler. And Milton Wexler was married to Nancy's mom, and he divorced her. He was a psychotherapist in Los Angeles. And it turns out that she was having chorea, so she was having involuntary movements, and she was having a very significant disease. And what Milton did is he had a lot of interactions with movie stars in Los Angeles, and he started the Hereditary Disease Foundation. And that foundation set about to figure out the cause of Nancy's mom's disease. And what Nancy did with some very special scientists to my heart, like David Hausman at MIT, is they went and identified indigenous populations in Venezuela and South America that had a very high incidence of Huntington's disease. And what they did is they analyzed, this was in the 70s and 80s, is these people who were not very educated about science were donating their tissue to Nancy and David and were doing analysis. And they developed technologies that were able to do the first mapping of a gene onto a chromosome in the late 70s, and they found the gene that causes Huntington's disease. And that's because Nancy was not going to take no for an answer. And she is not a geneticist. Nancy is a scientist. She's a psychiatry professor at Columbia, but nevertheless worked to find the gene. And only in 50 years from that Hereditary Disease Foundation, last year did an antisense oligonucleotide target and decrease the level of Huntington protein in a patient that had Huntington's disease. And so one thing I want to press upon you here is that a lot of these patient foundations, even though you don't get a payoff immediately, having the guts to go into the unknown and figure out what's going on can have very significant implications down the line. Now I'm going to talk about myotonic dystrophy, which is the most common adult onset form of muscular dystrophy. This disease is caused by a large and toxic repeat of CUG that's in a non-coding region of a messenger RNA. So this is the coding region of a messenger RNA, and this is the non-coding region. And smack dab in that non-coding region is this huge trunk of repeating RNA. So it's CUG repeats over and over. And if you have a repeat length of 50, that RNA forms this structure and causes disease. And so how the disease is caused is this long and toxic RNA becomes a high affinity binding site for this protein that's called muscleblind. And muscleblind protein actually regulates the processing of all of our genes. So the way that our messenger RNAs are made from our DNA is you get you get your DNA that gets encoded in a pre-messenger RNA. That pre-messenger RNA gets processed into a mature messenger RNA that makes a protein. Muscleblind controls the processing of that pre-messenger RNA. And when muscleblind, it regulates processing of hundreds of these. When this protein is not free to do that regulation, you have these diseases. And so these patients have issues of myotonia. And so that's, for example, if you drop a pencil into their hand and you ask them to squeeze it and let it go, they can't let it go. They have a myotonic discharge. And they can have other significant issues. That's all due to this toxic RNA-protein interaction. And so what we've been able to do is design compounds that can bind to this RNA target very specifically and kick off this bound muscleblind protein to make it now freely available to improving those splicing defects. The other thing that we've been able to do is make this molecule seek out and destroy or degrade that toxic RNA. Now, this degrader is the work of Ali Angebello, that's a graduate student in the lab. That degrading molecule, and these are all results that are in a preclinical mouse model for myotonic dystrophy. And so what you can see is we can degrade about 50% of this toxic RNA with this compound. Each one of these dots are one of those pre-messenger RNAs that are messed up when you have this disease. So all those dark dots are messed up. When we add our drug, 97% of those dark dots actually get improved. And the reason that you have this myotonic discharge in these patients is because you do not express a chloride ion channel protein on your muscle fiber. So basically what happens is when you have a contraction or relaxation in your muscle, you control energy. So you control the flow of energy between that muscle. So you contract it and you let it go. And so there are proteins that control the flow of ions to control the energy that's built up and let go as you contract and relax your muscle. That can include your hand and also your heart. Your heart contracts and relaxes all the time. And so here is, if we take a muscle fiber in that mouse, if it's a healthy mouse, what's colored in green is that ion channel that controls contraction and relaxation. You can see in DM1, you don't see any green because that protein's not there from these mice. However, when we deliver this degrader into these animals, we can see a very robust and significant upregulation of the expression of that protein. What we've also been able to do, and this is some work by Amy Carpenter that works at a startup company that Kevin Forrest and I started called Expansion Therapeutics. And so this is what the mice look like. So this is the untreated mouse. And so what you can do is you can pinch its hind legs, and its hind legs will have a cramp as a myotonic discharge in these mice. And so this is Amy's hand pinching the back of the mice. And so it's going to go to slow motion here. And so what you can see is the legs are cramped, right? It doesn't relax those cramped legs. When we give them our drug, you can see that the mice are moving around much faster in this cage. And when you're pinching on their hind legs, you can see that cramping is relieved. Here's a slow motion here. This is, so I think if I were to ask myself when we started out on this journey, if we would ever get to this level, I would have laughed in your face. No way on God's green earth would I think, one, I would have been asked to come to a place like Scripps, and two, would Scripps and the people in the lab have enabled me to get this far. There is no way I would have been able to say this. I could, fib a little bit and say, oh yeah, we would have done it. No way, okay? And so one thing is, what I'm telling you here are these molecules that we make in lab are tool compounds. They're not medicines, but we're hope to getting there. And so what we have to do now with these compounds, and we started Expansion to be able to do that, which I'll talk about later, is we need to design more drugs. And so one of the things in drug discovery that you have to do is you need to make sure a drug works, but also that it's safe. And so the way that you do that is you have this cyclical set of experiments where you take your lead medicine and you optimize it for having its effect. And then what you ultimately do is you do clinical experiments. And when there are things like rare diseases, it's very important that you have enough patients to be able to do a clinical experiment with enough data to know if the drug works or does not. And it's very important. So you need to ask the question, is your drug the first or best? Can you measure drug activity in a patient? Are there not enough patients to do a clinical trial, et cetera? And to get prepared to this talk, I did a couple statistical analysis. So it turns out that if the incidence of DM1 is the same across the globe as it is in the United States, 1 million people on Earth have myotonic dystrophy. That to me doesn't seem like a very rare number. Furthermore, on aggregate, on NIH's website, this number blew my mind, is that 1 in 10 people, 1 in 10 human beings has a rare disease. And I would say that you need rare people to solve this problem. You need patients to donate their tissue to say, I'm going to help this problem. And so I would say all the work that any of us do in disease biology is well routed in patients. I mean, God, we use HeLa cells in a lab. I'm from Baltimore. Henrietta Lacks, her HeLa cells were taken from her at Johns Hopkins University, right? And we're going back to bioethics now is much more evolved today than it was early on. And she had cervical cancer, so those cells are growing all over labs across the world. These rare patients are donating their DNA, their tissues. You're able to analyze those samples and figure out what's the gene that's causing the disease, how does the gene manifest itself. And so from this work that we were able to do at Scripps, we were able to start a company called Expansion Therapeutics. Suzanne Zuchek works in that company. She's a lead scientist. She was a postdoc in my lab. Also with Sai Velagapudi. Kevin Forrest, who's in the audience, is the CEO. Him and I started this company, along with Scott Rockledge. One of our partners in this is Eric Wang. Eric is a very important scientist in the DM1 area. His family actually has myotonic dystrophy. And that's very clarifying for us because it really shows us that our goal for this company is not necessarily to build up a technology and not have a real therapeutic. It's, they call these platform companies where you don't really make a medicine, you just flip a platform to a company. So we're very focused on taking these lead compounds and make them into a medicine. And we've also been able to work with really amazing people like Scott Rockledge. John Diekman and Scott worked at 5AM Ventures, which was the founding venture firm for Expansion. Elliot Eric, who is our chief medical officer. All of these are experiments. Amy Carpenter and Tim Blizzard are scientists at Expansion. And so what's really been amazing to me is I think we have a really important, when we first did these experiments, they were just basic science, but then as you start to get data and you go to these patient conferences, the data starts to make the experiments a responsibility, right? And the responsibility is these patients don't have any therapeutic. The responsibility is to see if you can work really hard and turn them into a medicine. And so I'm very grateful from all the people that have supported us to be able to do that. I just want to also end here by thanking the lab and thank you guys for your attention. I'm happy to answer any question. So does anybody have any questions? The microphones are coming around if you raise your hand. Yeah, it's hard to see. Yep.
It's likely that everybody here knows who Henrietta Lacks is, but
Yeah.
you mentioned her, and I wanted to impress that
Oh, I agree with you.
the
Yeah.
cells that were stolen from her without her knowledge,
Yep.
and then for decades, no recognition or honor was assigned to the family at all. Most of the people died in poverty.
Yep.
So I just think that's very, very important that we
It is.
stress that.
Yep,
Thank
I
you
agree
for
with you.
bringing it up.
I agree with you. That's part of the reason why I brought it up, because now we really need to be thinking about things in a bioethics manner for all of these tissues that these people are giving. They're their tissues. And so they have to have consent for how they're used. It's very important that we think about that. Yep. Any more questions?
I had a question on the, when you have these metastatic cancer cells that are already spreading, I understand that they're kind of sticky.
Mm-hmm.
And they've found some way to make them so they're not sticky, so they can't plant themselves in other parts of the body.
Yep.
How do they make them unsticky?
Yeah, so these molecules actually reprogram. They target a non-coding RNA and they reprogram these cells to not basically have invasive characteristics. So we shut off their pathways that cause them to be invasive in the first place. So that's how they work. So it's not that they're sticky, it's that there's an RNA that's upregulated to cause them to want to become metastatic, to go into the bloodstream, and then to shut them off from doing that.
You use the term non-coding RNA. is that RNA that functions similar to a protein and actually interacts with a receptor, or how does
Yeah,
that?
so a non-coding RNA by definition is an RNA that does not encode for a protein. So that's what I meant by non-coding RNA. Yep.
So when you target RNA, what is the quality of control that you get? How does that compare to if you are targeting a protein directly or you're targeting DNA? Is it a different type of regulation that you get from that kind of control?
It depends on the RNA. I mean, some of these non-coding RNAs can control the expression of many proteins in a single pathway. And so that one RNA will affect a pathway, but that RNA will have a synergistic effect to affect many proteins within that pathway to amplify its signal. And so it really depends on the target. Ones that amplify their signals within a pathway, we can very specifically target. And actually, the molecule signal gets amplified because it's affecting many proteins by targeting one RNA that's in that pathway. So it really depends on the target.
Have you done work on chronic fatigue caused by Epstein-Barr virus?
We have not, but Epstein-Barr virus has a lot of very interesting RNA structure. We've looked at it.
That would be much appreciated. It's a field that people have not gotten into.
Yep.
Because no big, well, there have been big celebrities with it, but it would be great if you did work on that. I would really appreciate it.
Right. Yeah, there's one of our collaborators, Walter Moss at Iowa State, actually has solved all of the structure within EBV, Epstein-Barr virus, and we've actually looked at it in a very significant way to look and see what therapeutic targets there are for it.
I appreciate it very much. If you could.
Yep, we'll look at it more. Thank you.
Matt, have you been able to profile the expression of genes that are being altered when you hit it with a drug that is giving you a positive phenotype, and then see anything in it, in that profile, that says, aha, this is what's really driving the rapid cell division or whatever?
So we have worked on making HER2 negative cells HER2 positive, and we've been targeting non-coding RNAs broadly and sphingosine 1-phosphate receptor, where we've gotten some insights into like how invasiveness in cancer cells is turned up for some protein targets. So we have some data to support that you can do that, but not a lot. That's going to require us to, I think, do full RNA-seq and full proteome with and without the drug.
Full RNA-seq on these, that's exactly.
Yeah, we
don't
do
have
that.
that data right now.
We do that routinely.
It's a massive.
Yeah, we do that
And
routinely.
you got to do it at some point.
We do that. Well, we do. That was full RNA-seq that I showed you in the mouse muscle. But we routinely do full RNA-seq and full proteomics when we treat with these drugs to whittle down what is the specific, and we have a whole pathway for what is the specific gene that's targeted, how do we validate that that's modulating the phenotype, et cetera, right? So there's a lot of discovery biology that we can do here on the back end.
Yeah. In some of these tumors, when you see the profile, you see one or two things in there that very clearly are associated with rapid cell division, for example. And I was asking more specifically whether you can pick out things from with drug, without drug, where you can see things that really seem to be tightly correlated with the phenomena you're looking at. It's a tough one, but I mean, I think it's obviously where a thing has to go at some point.
So we do have, I don't think we've looked at it at the level that you're wanting us to have, and now because Paul wrote the textbook in biophysical chemistry, I'm going to now stare at these on my flight home.
And we have Turner's rules, your mentor's rules in that textbook.
Yes, I know. Doug was very happy. Sorry? Okay, I have time for one more question, and then we should go to the reception.
I wondered what the status of this science was in the treatment of Alzheimer's disease. Have you done any work with that?
We have done Alzheimer's. My mother died of Alzheimer's on Christmas Day of 2015. And Christmas was her favorite day of the year at 9 o'clock in the morning, right after us kids would have probably opened up all of our presents. I'm telling you, Mom, that was her day. So we are working very, very hard on Alzheimer's for Mom. My daughter's named after my mother. And so there are many, when you have Alzheimer's disease, there are many genes that are upregulated that cause inflammation or damage to your brain, and many of them are due to RNA. There are many RNA dysregulations that are due to that. I actually got my mom's cerebellum is stored in liquid nitrogen, so I was hoping a day when I could sequence her genome and figure out what the targets are. You're working very hard on that. That was very important to me to get that. So, thank you. We'll have a reception outside.